Good morning, students. Today we discussed about a new chapter, that is strategies for enhancement in food production. Third botany chapter in plus 2.
In this chapter, the first topic is animal husbandry. It is the agricultural practice of breeding and raising of livestock. 70% of world's livestock are in India and China. In this chapter, we mainly discussed about dairy farm management, poultry farm management, beekeeping, fisheries, etc. First, dairy farm management. Dairying is the management of an animal for milk and its products for human consumption. Milk yield is primarily dependent on the quality of breed in the farm. Cleanliness and hygiene is also required for good milking. Machine work, machine work helps to reduce the problem. That means to avoid contamination, we must change the procedure from hand milking to machine milking. Adequate consulting of doctors with cattle is essential because many diseases are present. Foot and mouth disease, inflammation of udder, anthrax, etc. are common cattle diseases. Anthrax is a disease that transmits from these cattle to human and it is a dangerous disease. Milk is a balanced diet as it contains sugar, protein, fat, minerals, vitamins, etc. And milk sugar is known as lactose and protein is known as casein. Dr. Varghese Kurian is known as Milkman of India and he is the father of White Revolution. And White Revolution starts at Anand in Gujarat state. And he started a cooperative society known as Amul. And the full form of Amul is Anand Milk Union Limited. We know about various products are now also available in market of Amul's product. Next one is poultry farm management. It is the class of domesticated fowl, birds used for egg or meat. That means we know very well about some birds called chicken and like kada, etc. These are mainly cultivated for egg and meat. Proper selection of bird or breed, proper hygiene, etc. are essential for this practice. Disease free selection is an important criterion because many of such birds are collaboratively or collectively cultivated. So, if one has infected, there is no social distancing. So, all of the farm are infected. Avian flu is a viral disease caused by virus H5N1. Newcastle disease is also a viral disease. Coryza, Pasteurellosis, Salmonellosis, etc. are bacterial disease. These avian flu are very common to us because it is affected some birds in Kozhikode in the recent time. Beekeeping. It is the maintenance of hives of honeybee for the production of honey. Hives means the living situation of honeybee is known as hives. Honey has high nutritional value, medicinal property, and so on. Apis indica is the most common cultivating species and Apis mellifera species are also used. Then we move to fisheries. This industry devoted to the catching, processing, or selling of fish, shellfish, or other aquatic animals. Blue revolution is related to fish resources. That means we know that the color of ocean due to scattering is blue. So blue revolution is related to this field. Katla, rohu, tilapia, mrigal, etc. are freshwater fishes. Sardine, mackerel, prawn, crab, etc. are marine. Sardine means matti. Mackerels means ayala. Prawn നമ്മുടെ ചെമ്മീൻ. Then there are some confusing factors. That means aquaculture versus pisciculture. Aquaculture means it is the saltwater or freshwater breeding, rearing, and harvesting of aquatic flora and fauna for commercial purposes. That means it includes all the field in fauna. Fauna means aquatic animals, flora means aquatic plants for commercial purposes. So and also it include freshwater as well as marine water. So it is the whole field it include. While in pisciculture it is just culturing of fish or fish farming to obtain fish products. So it aquaculture contain all the field while pisciculture has a uh, small area of aquaculture. Here is a chart showing animal breeding procedures. And breeding procedures has two branches, inbreeding and outbreeding. Outbreeding again converted as outcrossing, crossbreeding, interspecific hybridization. Let us look into inbreeding. Inbreeding means breeding between animals of same breed. Through inbreeding, we can obtain pure lines, while the chances of inbreeding depression is more. That means we know about Gregor Johann Mendel. He produced pure tall tree and pure dwarf trees. He obtained this pure tall tree by this procedure called inbreeding. Pure tall tree has capital T and capital T, and pure dwarf tree has small t and small t, that he obtained through this procedure. While the chances of inbreeding depression is increases means inbreeding depression means the production of inferior quality products. The continuous inbreeding result the chances of inferior quality products production. So this inbreeding has some limitations and also some benefits. Next is outcrossing. Crossing of animal within the same breed but having no common ancestor on either side of their pedigree. Pedigree means their ancestral chart up to 4 to 6 generations. The offsprings are called outcross. For example, a dog called German Shepherd. The male German Shepherd is from one country and female German Shepherd is from another country. Both these German Shepherd does not have same common ancestor up to 4 to 6 generations. And if we crosses these two German Shepherds, we obtain a new offspring, and that cross is known as outcrossing. These two German Shepherds are in same breed but does not have same common ancestors. Then crossbreeding. Here, superior male are crosses with superior females of another breed. So different characters can be joined together in offsprings. Here also we can say an example by using in the case of dog. We know a category of dog called German Shepherd and another breed called Dalmatian. These two dogs are from various countries, and if we crosses these two dogs, we obtain a new product. That new offsprings has both the characters of the superior male and superior female. That type of breeding is known as cross breeding. In NCERT text, there is an example. Hirserdale is a new breed of sheep. This Hirserdale is produced by crossing two different breeds of Beccanary Eves and Merino Rams. In this picture, the first two are Beccanary Eves and Merino Rams. These two are from different breeds, and we obtain a new breed, that is the offspring. These offsprings as both the characters of their parents. Next one is interspecific hybridization. Here, two unrelated species in a genus are crossed together and produces a new offspring. In this picture, we can see the crossing between horse and donkey. Horse and donkey belongs to same genus while the species name are separate. We can cross these two different species, and here we produce mule by crossing horse and donkey, while this mule or the offsprings produced through interspecific hybridization are sterile. This is the main drawback of interspecific hybridization because the nature limit this type of crossing. A hinny is a domestic equine hybrid, that is the offsprings of a male horse and a female donkey, and it is the reciprocal cross to the more common mule, which is the products of a male donkey and a female horse. That means hinny and mule are two varieties produced through this interspecific hybridization. The liger is a hybrid offspring of male lion and a female tiger. That means this liger has more attraction and strength than tiger and lion while they are sterile. A tigon or tiglan is the hybrid offsprings of male tiger and a female lion. These tiger and lion belongs to same genus Panthera, while the species name only changes. In the case of tiger, the species name is Tigris, and in the case of lion, the species name is Leo. This type of crossing is known as interspecific hybridization. There are some procedures for the breeding of animals. First one is artificial insemination. The semens collected from Male are stored in minus 196 degrees Celsius called cryopreservation. In cryopreservation, we can store semen, germplasm, ovum, etc. without damage, and whenever needed, they can be used. So, whenever this semen is needed, they transfer to female uterus. This practice is mainly adopted in the field of cattle. Here, some superior quality male sperms or semen is collected and cryopreserved. And whenever the estrous cycle of cattle, female cattle taken place, this semen is transferred to their uterus artificially, and that process is called artificial insemination. Through this practice, good quality offsprings can be produced. Then next one is MOET, multiple ovarian embryo transfer technology. It is a method in which hormone induced ovulation in female parent and such egg are collected, then artificial fertilization done, then transferred to several females, that means surrogate mothers. Here is a picture, that means through MOET, by applying hormones, several ovums are produced from a female parent, and they are artificially fertilized outside the body and cultured. Then transfer this cultured embryo into several surrogate mothers, so that these surrogate mothers only carry this embryo and several offsprings can be produced. That means this is a technology to produce good quality products. And first one is artificial insemination, and the second one is MOET, that means multiple ovarian embryo transfer technology. These technologies are mainly participated or done in the case of animals. Then we move to the field of plants. Plant breeding improve crop quality, yield and economic benefits. Norman A. Borlaug is considered as the father of green revolution in world, and the origin of green revolution is at Mexico. Green revolution means a drastic improvement of crop quality through breeding procedure is known as green revolution. In India, Dr. M.S. Swaminathan is known as father of green revolution. There are various stages present in plant breeding procedure. First one is collection of variability. The entire collection of plant or seed having all the diverse alleles for all genes in a given crop is called germplasm collection. That means in this procedure we collect all varieties of a species called tall plant, dwarf plant. If fruit have different colors, so one is yellow, one is red, and etc. So we collect all these colored varieties, and seed coat have different varieties, so we collect all the seed coat different varieties, and so on. That means all the diverse characters in a species are collected here. In some botanical gardens, if we visited, we can see same species have a same species is conserved there with diverse characters. That preservation is known as germplasm preservation. Then we move to next stage, that is evaluation and selection of parents. Here, pure lines are produced with desirable characters. That means here we implement the technology that is inbreeding that we mentioned earlier. Gregor Johann Mendel produces pure breeds through inbreeding. Here also we produces pure breeding through that technology. Then the next stage is cross hybridization among the selected parents. This cross hybridization has some sub-stages, that is emasculation, transfer of pollen grains from male to female, bagging, tagging, etc. These subdivisions are studied in sexual reproduction in flowering plants in the previous chapter. So these stages are done in the third stage called cross hybridization. Then the fourth stage is selection and testing of superior recombinant. Through this technology, we select the superior recombinant. That means by recombination or cross hybridization, various offsprings are obtained or many offsprings are obtained. Some of them are superior, some of them are medium, some of them are inferior. We only select the superior quality plants only and again closed together and eliminate the inferior quality from that plant group. So this selection and testing of superior recombinant is a crucial stage. Through this stage only we can obtain the superior quality products by eliminating the inferior and bad qualities. Then we move to the fifth stage, that is testing. It includes testing in several seasons. That means one variety should be commercialized only after testing within different season, because India is a country having different kinds of season in different localities. So only benefit is obtained when it is cultivated in different season. Then different soils, that means India having different kinds of soil. Then releases and commercialization of new cultivars. That means after this all testing, we releases this product into our society or in the field of agriculture. Mainly it is done by government itself, while some of the private agencies also done these procedures. Many plant breeding stations are present in India across. This plant breeding station is controlled by many scientists and project associates and project assistants, etc. They have the duty to cross hybridization testing, etc. in this condition and also produces new varieties for future. There are many objectives for plant breeding. Some of them are high yield, resistant to disease and pest, changes in maturity period, elimination of toxic substances, synchronous maturity, photosensitivity, resistant to abiotic stress, improved quality. Some more objectives are present and the main objectives are mentioned here. and through this technology, that means plant breeding technologies, and we produce some crops. First among them are wheat and rice. We produce semi-dwarf varieties of wheat, and their names are Sonalika Kalyan Sona. They are high yielding and disease resistant. Their picture are mentioned below. IR8 is a variety of rice developed by IRRI. That means IRRI is a short name of International Rice Research Institute, Manila, Philippines. So this International Rice Research Institute produce this IR8. It has very important role in the field of agriculture. Then Taichung native 1 is a species produced locally by Taiwan government. Later Jaya Ratna are produced in India itself. These Jaya Ratna etc. are now also available in market. Then the next one is sugar cane. In the case of sugarcane, there are two varieties, Saccharum barberi and Saccharum officinarum. Saccharum barberi is a North Indian variety. It has poor yield and less sugar content, while Saccharum officinarum is a South Indian variety. They have thicker stem and high sugar, but not suitable for North India. That means some genetic factors allows this Saccharum barberi in North India and Saccharum officinarum in South India. So in order to obtain this Saccharum officinarum's quality in North India, the plant breeders crosses these two varieties, that means Saccharum barberi and Saccharum officinarum together through the earlier discussed methods. That means five methods are discussed in earlier. In such methods, the Saccharum barberi and Saccharum officinarum are crossed together. By crossing these two, we obtain a new variety of thicker stem and high sugar containing sugar cane that is suitable for North India. Then in the case of millets, millet also produced through Green Revolution. 2018 is considered as the International Year of Millets by United Nations. Hybrid maize, jowar, and bajra have been successfully developed in India. Hybrid breeding has led to the development of several high-yielding varieties resistant to water stress. In India, we know that water stress is a problem. So, to overcome this problem, by artificial hybridization and breeding procedure, we can produce plants with resistant varieties for water stress. Then through these procedures, we produce many plants for disease resistant. Here are some plants with various kind of disease resistant. In the case of wheat, the variety's name is Himgiri. They are resistant to leaf and stem rust, hill bund. We know that Rust is a disease caused by fungi. So, such a pathogenic effect can be decreased by crossing different varieties together. Then, in the case of brassica, the mustard, that means കടുകൊക്കെ ഉൾപ്പെടുന്ന ഫാമിലിയാണ് ബ്രാസിക്ക. Brassica Pisa sornium is a variety. It is resistant against white rust. Then in the case of cauliflower, Pusa subra, Pusa snowball K1, etc. They are resistant to black rot and curl blight black rot. Then in the case of cowpea, it is a kind of pea plant. The new variety is Pusa comal. It is resistant to bacterial blight. Then, in the case of chili, the variety's name is Pusa Sadabahar. It is resistant against CMV, that means chili mosaic virus. TMV, that means tobacco mosaic virus, and leaf curl. And many of having a doubt, what is Pusa? Dr. Rajendra Prasad Central Agricultural University, formerly Rajendra Agricultural University, is an agricultural university located in Pusa. That means these all varieties, Pusa Swarnium, Pusa Subra, Pusa Komal, Pusa Sadabahar, etc. are the breed that produced from this university. So that is why they get a name called Pusa. Earlier we discussed about a rice variety called IR8. These IR represent the breeding station, that is IRRI, International Rice Research Institution. Likewise, Pusa is the place where this Central University, Agricultural University is situated. Then we move to the next breeding procedure, that is mutation breeding. We know that mutation produced by alpha, beta, gamma, X-ray, UV rays, etc. can change genetic variation. If genetic variation is favorable, it is a good change. So by mutation breeding, we must encourage or we must change their genetic variation for good purpose. It is the process by which genetically modification of crop by applying radiations like gamma radiation. Gamma garden is a mutation breeding setup. Layout of gamma garden. Here in the central portion, it is the radiation source. Then the sector 1, sector 2, sector 3 wall. There is a thick concrete wall surrounding this gamma garden because gamma radiation is a hazardous gas. If it is escaped to outside, it may lead to mutation for animal also. So in sector 1, sector 2, sector 3, same plants are arranged and study the changes that has taken place through the application of gamma rays. If it is favorable, that plant breed together and produce new varieties, and if it is unfavorable, they are destructed. This type of breeding procedure is known as mutation breeding. Then through breeding procedure, the plant breeding for developing resistant to pests. Earlier we discussed about the plant breeded for disease resistant. Here we discussed about resistant to pest. First one is brassica. Variety's name is Pusa gaurav. It is resistant against aphid. Aphid means a small insect. Then flat bean, Pusa sem 2, Pusa sem 3. It is resistant against jascides, aphids, and fruit borer. These three are small insects which nourishes from the stem and fruits of this flat bean. So by cultivating this new variety, we can avoid this problem. Then okra. Okra means Our Lady's finger. Variety name is Pusa Swarnium Pusa A4. It resistant against shoot and fruit borer. Then we move to plant breeding for food quality. Here in this chart, that means the map of world, the red colored areas are threatened by food scarcity and green areas are developed countries. So the threatened countries are many in world. The major threatened area is in this picture is Africa and other many countries like India and so on. So, in order to avoid this food scarcity, we must improve the food quality through these plant breeding procedures that can be discussed in the following charts. Biofortification. It is the breeding crop with higher level of vitamin and minerals or higher protein and healthier fat to improve public health. Their objectives are protein content and quality, oil content and quality, vitamin content, micronutrients and mineral content. Maize that had twice the amount of amino acid, lysine and tryptophan. Atlas 66 is a wheat with high protein donor for improving cultivated wheat. That means through this biofortification, we can improve the crop quality by adding various nutrients. This also obtained through the earlierly discussed plant breeding procedures. By biofortification, we can improve the public health. Next one is single cell protein. Short form is SCP. Microorganisms produce more protein than larger organisms compared to their biomass. These can be utilized for diet. It may reduce poverty, good effect on landless cultivation, etc. Because their cultivation need only laboratory itself. We know about chlorella, spirulina, etc. This chlorella and spirulina can be utilized by space travelers itself because this chlorella can be cultivated by the space shuttles itself. Only the light from the bulb required for the multiplication of chlorella. And chlorella and spirulina capsules are now available in market. Then yeast cake. Yeast can be multiplied by applying sugar content. So yeast is also a SCP. Then we move to tissue culture. Tissue culture is the another way for enhancement in food production. Here, totipotency is the principle of tissue culture. Totipotency means the ability of an individual cell to become whole organism. In this picture, the first shown image is a cluster of cell produced from a single cell. This cluster of cell is known as callus. From this callus, we can cut small cells separate, and by applying hormones, we can produce rooting, we can induce rooting and shooting. And in the third figure, root and shoots are initiated. So from this callus, we can produce thousands of plants, and this process is known as micropropagation. So we required a nutrient media for this callus cultivation, and most preferably used nutrient media is Mirage and Squeeze medium, or short form is MS medium. It contains a carbon source, water, minerals, vitamins, hormones, amino acid, salts, etc. This medium is invented by two scientists called Mirage and Squeeze. That is why this medium has this name. This is a simple image of already prepared Mirage and Squeeze medium that we can readily used for readily used as nutrient medium. Then micropropagation, the process of propagation of thousands of plants through tissue culture is called micropropagation. That means from a single cell itself we can produce thousands of plants. This can be explained using this image. Here in this image there is a mother plant. From this mother plant we can collect some cells and transfer to nutrient media. So in this nutrient media it produces callus formation. Callus formation means multiple cells formation. Then we cut this callus into small pieces and subcultured this callus, because the already cultured medium is depleted due to the usage of nutrient media by the cells. So they get subcultured. Then we apply hormones for these subcultured mediums so that rooting and shooting can be produced. Then these all procedures are done in laboratory, so that there is an acclimatization problem. That means from laboratory to field there is a chance of climatic change. So primary hardening is required. These primary hardening are mainly done in poly houses or in some like this polythene polyhouse gardens. Then we move to secondary hardening. In secondary hardening, these are these plantlets are moved into small fields for acclimatization of these plantlets to the external environment. This can be discussed in the next image. Here there is a plant, mature plant. First one is a carrot plant. Wash the carrot plant thoroughly. Then break into 3 or 4 pieces. Then using a cork borer, we cut a small piece, and this small piece is used as a cluster of cell and transferred into this medium, nutrient medium, so that it produce cluster of cells called callus within 4 to 6 weeks. Then we cut these callus into small pieces again for subculturing. Then subculturing means these small piece of callus are transferred into new nutrient media for further cell division. Then we add hormones for the initiation of rooting and shooting. After 4 to 8 weeks, rooting and shootings are initiated. That means root and shoots are initiated. Then primary hardening, secondary hardening, etc. are done to acclimatize these plantlets for the external feed so that from a single plant we can produce thousands of plants, and this technology is known as micropropagation. And from a single cell itself many thousand plants can be produced. That is why this technology get the name tissue culture. Tissue means a group of cells, culture means cultivating the tissues. That is why the name tissue culture. And tissue culture and micropropagation are included in same area while they have some difference. In viral or bacterial infected plant, the meristem can be utilized for tissue culture. Meristem means the newly produced cells, meristem, root apical meristem, shoot apical meristem, that you have studied in earlier classes. So this meristem does not infected with this virus or bacteria because they are newly produced cells. So these newly produced cell can be utilized for tissue culture in such plants, and through tissue culture we can produce the same plant of their same character genetically and morphologically same plant of parent. That means morphologically and genetically similar to parent, that we called as clones, carbon copy of an individual, that means parents. This tissue culture is commonly done in plants. This tissue culture can be done in animals, but the practice of tissue culture in animal is restricted by laws. Okay, I hope all of you understood the topics that we discussed in this chapter. We can see in the next class with a new chapter.
Thank you for watching this